Hello everybody, welcome back. In this video, we're going to talk about neuronal activity. So we're going to talk about the neuron, we're going to talk about glial cells, and we're going to talk about how they communicate with each other. I don't think we're going to get into the action potential on this video. I'll probably do another short video to talk about the action potential because I'm trying to keep these videos nice and short, a little more palatable to watch. So let's go ahead and get started. Neuronal activity. Sensory neurons send messages from the sensory neurons to central nervous system. So the sensory neurons, so if I'm touching the desk and I feel that it's nice and cool, I have sensory receptors in my fingers, and these sensory receptors will send a message to the central nervous system by using sensory neurons. So the receptors pick it up, a little magic occurs. It's a kind of complicated thing, but there's a process that occurs that translates this energy from touching the desk into the type of energy that is used by the neuron, so that this energy from the desk is translated to an impulse, which can be sent then to the central nervous system. Motor neurons send messages from the central nervous system to muscles. So when I need to move my leg, my central nervous system will send a message out, and then I can move my leg. Glial cells are a type of cell found in the nervous system that serves a supportive role to the neuron. So basically, they are structurally supportive by helping the neurons stay in place. They are also helping to remove waste. They're helping to provide nutrition. And there is some newer research that shows that some types of glial cells can actually help with the communication between neurons. So they do a very large role, and we'll talk about another couple of roles that they have also in a few minutes. Interneurons convey messages between neurons in the brain or spinal cord. So remember when I talked about our reflexes in the previous video, I talked about how, if I'm going to touch the hot stove, the message goes like my sensory neurons goes from my receptors in my fingers to a sensory neuron up to the spinal cord, and my spinal cord says, oh no, we're going to fix this immediately, and sends a signal back down. It doesn't go up to the brain. Instead, it uses an interneuron to connect that incoming signal to that outcoming signal. So that's an example of how the interneuron works. Here is one of our best friends in the world, the neuron. I love it so much. Okay, let's take a look at the structures of the neuron. We're going to start down here at the bulky end. Just like any other cell, the neuron has a cell body, which is the soma, which is this area around here, and it has a nucleus. This ball thing here is a nucleus. With this particular illustration, it almost looks like the arrow is pointing to the nucleus where it says soma. It's kind of a poor placement of that arrow. This arrow would be better suited pointed down here somewhere, because the soma is this cell body. It is kind of the bulk of the cell, whereas the nucleus of the cell is where our DNA and all that interesting material is at within the cell. But we're not going to get into all that right now. The branch-like structures that we see that look like little trees with no leaves, those are called dendrites. The purpose of the dendrite is to receive signals from other neurons. So this is basically the receiving end of the neuron. A signal will come in from a different neuron, stimulate these receptors on these dendrites. These dendrites have a little thing on them called a dendritic spine. And on these little spines are receptors. The receptors receive the neurotransmitter, which is the chemical messenger, from the upstream or presynaptic neurons. So the neuron sending a message over here somewhere would release a chemical called a neurotransmitter. That neurotransmitter would float over here and bind to one of these dendritic spines that has a receptor. So it'll bind to that receptor and cause changes within the cell. if enough change occurs in this cell, and actually what's happening is it's changing the electrical voltage of the cell. So it changes the electrical voltage of the cell. When it gets enough stimulation on these dendrites, then a signal shoots down this cell all the way to the end. This section here is called the axon. The axon is where that signal will shoot down through. So if we have a lot of electrical stimulation, a lot of stimulation here by the chemical messenger. So these dendrites receive a lot of signals, then changes in the electrical charge of the inside of the cell gets to a certain level, and bammo, it sends a signal down to the end of the neuron. That signal goes right down this axon to the end, to these to what's called the terminal. The terminal is the end of the neuron here. And at the end of the neuron, we have synaptic terminals. They can also be called synaptic buttons. And so this is where this neuron, if it was going to send a signal to a different neuron's dendrites, it would release its chemical messenger here, which would float across and meet somebody else's neurons over here, somebody else's dendrites. So this chemical message would be released. The dendrites of a next neuron downstream would pick up that message, and then the cycle would continue. So the main part, one more part that we have to talk about that's super important is the myelin sheath. Now, especially in the nervous system, and especially with our motor neurons in our peripheral nervous system, the myelin sheath is very important. Myelin is kind of a fatty substance. It is physically created by glial cells, which are those cells I talked about that are kind of helper cells to the neurons. What can happen in the central nervous system is one glial cell can wrap itself around several times on this neuron and create a myelin sheath. So these little pieces of myelin right here are acting as a superconductor, or not a superconductor, but they're an insulator. For example, you know how electrical wire has that plastic on the outside so that the electricity stays within that wire and travels really fast? Well, this is what the myelin sheath does. It helps the signal that is being sent from this cell body down to these synaptic terminals. It helps that signal to move faster. With this myelin insulating, all that has to happen is the signal will go shoo through here, and then it'll just it'll pop through it just kind of hops. It hops instead of having to go all the way down. Okay, these little gaps in between the neurons are called the nodes of Ranvier. Nodes of Ranvier, and the nodes of Ranvier are where some of that potential will take place, and then it'll zip past, and then it'll take place, and then it'll zip past, and take place, and then zip past. So basically, it really speeds up that transmission of that signal. So once again, we have our dendrites, which are these tree-like structures, and their purpose is to receive messages from upstream neurons. So neurons that are releasing their chemical messenger called neurotransmitter are going to float across and bind to receptors that are on these dendrites. On their little dendritic spines, there's going to be little protein receptors. These receptors, when enough of them are stimulated, it is going to change the voltage of this cell because it has a voltage in it. We are creatures that have electrical voltage within us, believe it or not. When it gets to a certain level, the neuron will send a signal down the axon to the synaptic terminals, where it can then release its own neurotransmitter downstream. Okay, so we got our soma, which is the cell body. We have our nucleus. We have our dendrites. We have our myelin sheath, once again, that is to insulate and speed up the signal going down the axon. We have the axon, which is this tail-like part where the signal travels. It only goes one direction, down away from the cell body. And we have the synaptic terminals down here where this neuron would release its own neurotransmitter or chemical messenger. Okay, one brief thing I want to talk about with the myelin sheath. Myelin is super important. And there are diseases such as multiple sclerosis, I can't say it, multiple sclerosis, that attack the myelin. And this happens in the peripheral nervous system. I believe it happens in the brain too, but a lot out in the peripheral nervous system. I'm not super familiar with the disease, but I do know that it's a disease of the myelin sheath, where these myelinated neurons lose their myelin. So then the signaling starts to go very slowly. And that way movement is impaired. And that is something that occurs with multiple sclerosis. And it's called a demyelinating disease, meaning that it takes away the myelin. And there are other diseases that do this also, and they're pretty terrible diseases. All right, here's some terminology for you. The structure of the neuron: soma, cell body, neuron control center. So no, no. The soma is the cell body, okay? That is the cell body. The neuron control center would be the nucleus. So I'm sorry about this. This is a stock slide that I did not notice that error in. The dendrites are the branch-like structures that receive neural impulses. The axon is a single fiber that sends the neural impulses down to the terminal button. The terminal button is the place where the neurotransmitter is going to be released, the chemical messenger. The myelin sheath is the insulation that goes around the axon. And the synapse, which is a term I don't know if I've used it yet, the synapse is that gap between the terminal button where the neurotransmitter is getting released and the dendrites of the next neuron that are going to pick up that neurotransmitter. So that gap between them is the synapse. So it's interesting because the nervous system, it's not like a regular wiring system where you would like twist the wires together and make sure they're touching like a regular electrical system. In this case, the signal is sent through a chemical messenger between neurons from one neuron to the next. But once the chemical messenger is received by the neuron, within the neuron, there's an electrical signal that sends down to the end. So it is called electrochemical communication because we have an electrical signal within the cell and chemical communication outside of the cell. Here are the glial cells that I was talking about. In the central nervous system, a glial cell might look like this. It's an oligodendrocyte. So this oligodendrocyte, if you see here, we have a neuron. This is the cell body. We have the nucleus. We have the dendrites out here. Not the greatest drawing, but these are the dendrites out here. Then we have our axon that is going this way. And then we have our terminal down here with our terminal buttons on the end. So this one cell can provide the myelin. It's in this particular case, it's providing 4 pieces of myelin to this. We might have another cell providing these other ones, or maybe this cell could provide all the myelin for this particular neuron. The point is, in the nervous system, one glial cell can provide multiple pieces of myelin for a myelin sheath, whereas in the peripheral nervous system, one cell would be needed for each of these sections. So that's kind of a big difference in how the myelin sheath works in the central nervous system and the peripheral nervous system. And then these are some other type of glial cells. And they all do those jobs that I talked about with nutrition, the cleanup crew, all sorts of support roles like that we already talked about. All right, synaptic transmission. Remember, we talked about the dendrites getting stimulated to a certain level, and then if they get stimulated enough, the neuron sends a message down the axon to the terminal, to the axon terminal, or the terminal button. So this is a close-up view of that terminal button. And what we see going on here is we see that there are little packets of neurotransmitter in here. So it's little packets of a chemical called a neurotransmitter. And when this cell is stimulated to the degree where it fires its signal, called the action potential, that signal, these packets merge with the cell lining, and then they release their packet into the synaptic cleft. Okay, the space in here, which is the synapse, it's sometimes called the synaptic gap or synaptic cleft, or just called synapse. But that's the gap between the what we call a postsynaptic neuron, which is the upstream neuron that is sending down the neurotransmitter. And then this one would be this is the presynaptic, I'm so sorry. The presynaptic neuron is sending its signal, and the postsynaptic is receiving it. So these little dots are representing the neurotransmitter being released, and these little protein structures in the cell wall are the neurons that are the receptors. So these will bind onto there and trigger some sort of reaction within the cell. Okay, so once again, the signal comes down, these vesicles will merge with the cell wall, release their contents, which would be the neurotransmitter, which would then be picked up by the dendrite, by a receptor on the dendrite of the downstream neuron. So if this is a cell, what we would call a dopaminergic cell that releases dopamine, then it would release its dopamine into the synapse. This would have to be a dopamine cell also in order to pick up this neurotransmitter onto its receptor. So if this was a dopamine neuron releasing dopamine, and this dendrite that we see down here with the receptors on it is to a serotonin cell, serotonergic, that is only dealing with serotonin, then my neurotransmitter can't bind to these receptors. A dopamine cell will release dopamine, and then it has to be picked up by another dopamine cell. Okay, I'm calling them dopamine cells, dopaminergic cell. I'm just trying to keep it a little bit more simple. And so if this was serotonin, then it would have to be picked up by receptors on a serotonergic cell. If it was glutamate, it would have to be picked up by a glutamate cell. So in other words, the neuron that is releasing the neurotransmitter has to be the same as the one that's picking it up, if that makes any sense. I hope it does. All right. What else did I want to say about this? Okay, yeah, these are just little receptors. And what happens here actually is this neurotransmitter, once it's released, it binds to the receptor, which triggers a reaction of sorts inside the cell. So it triggers a reaction inside the cell, and then it releases from there and starts to drift off. And then what happens here is that we have all this neurotransmitter left in this synapse. What do we do with it, and how do we get rid of it? There's a couple of different ways that we get rid of it. There's three main ways that we get rid of it. The first one is called a neurotransmitter reuptake pump. And that would be another protein that's in the membrane of the cell. And what happens is it would suck up the loose dopamine that's in the synapse, bring it back into the terminal button, and then repackage it into one of these vesicles so that it could be used. Okay. Another way is that there are enzymes in the synapse. There are enzymes specific to the different neurotransmitters. So there is a specific enzyme that breaks down dopamine, if this is a dopaminergic cell. So it would break that dopamine down into its base components, which can then be reabsorbed and made into something else. The third way is diffusion, that the little molecules of the neurotransmitter will just drift out. So those are the three ways that that neurotransmitter is removed from the synaptic cleft. So once again, we're just going to kind of do a little bit of a recap here. This particular image, we have a synapse, which is actually on the cell body. That is something that can happen as well. Most communication is going to occur with the terminal buttons communicating with a spine on the dendrites. However, it is possible for a synapse to occur on the cell body as well. So what would happen here? We have our upstream neuron here, our upstream, or in this case, we would call it a presynaptic neuron. We have our dendrites up here. This particular neuron, we're going to say, okay, he already got stimulated. He already got a whole lot of neurotransmitter here, which stimulated this cell, caused changes within the cell that made that cell change its voltage, and then when it gets to a certain level, bam, it sends its signal. So it sent its signal down to the terminal buttons, and then here at the synapse, it's releasing those little vesicles, which are going to merge with the cell wall and then drift out into the synapse. And then receptors on the membrane of this other cell are going to be stimulated by that neurotransmitter, as long as it's the same type of cell. Okay, and then it would just keep on going on down the line. So some terminology: synaptic transmission. The synapse is the gap between the sending neuron and the receiving neuron. Mechanisms of synaptic transmission. The synaptic vesicles at the end of the axon release neurotransmitters. Remember, I talked about those little vesicles will merge with the cell wall and then release into the synapse. Neurotransmitters attached to receptor sites on the dendrites. Yep, a neurotransmitter molecule will bind to that receptor, cause a change within the cell, and then release it back into the synapse. This may cause an action potential to occur. Now, an action potential is what we call that stimulation that causes the neuron to fire. So when the neuron gets enough stimulation, then it changes its polarity, it changes its negative charge, it changes that, and then it shoots an action potential down that axon. And we'll get into details about that in our next video. So how are the neurotransmitters removed from the synapse? Reuptake, they're taken back up. Enzymes that break down the neurotransmitters, and diffusion. But we already talked about that, so I don't really need to go into a lot of detail on this. Okay, next video, we're going to talk about the action potential. We're going to talk about how the cell changes. Remember, we talked about the dendrites picking up the neurotransmitter, binding to the receptors on their dendrites, on those dendritic spines. And we said that the stimulation from that makes some sort of change occur within the cell. We're going to talk about the changes that occur within the cell, and we're going to talk about the mechanics of the action potential. We're going to get into that in our next video. Okay, that should do it for this one. I hope this is helpful to you guys. And if you have any questions, as usual, please feel free to shoot me an email and let me know your questions, and I'll see if I can clarify it. Okay, I can either do another short video or I can just talk to you over Zoom. Okay, all right, you guys have a good day. Take care. Bye-bye.